That's
All right.
You say because you were everyone else thought it was on
Okay. We are finished with the first part of it, which was the nucleus and the ribosomes, although they come up because what they actually produce, the proteins, get processed in the next part as well. It's so dark out. You want to, Aaron? Can you open that? I think.
It's gonna be dark
No, it's gonna make me cold seeing how cold it is outside.
Oh, it's not so bad. It's not so bad. We're gonna need,
You turn the heat on?
some more here.
Dang, I wish I could just turn out front of my hand.
No,
It's
I tried. It's too embarrassing.
there's a snake that actually killed me. I have to work on it.
Adrian, let's give it a shot and see what it's like with the light off, because I'm afraid that.
All the way.
Ooh, that's pretty
I
good.
can't read it.
No, it works fine.
It was worse than killing stockings for me.
No
It
way.
was so much
No
worse.
way.
Yes, it is.
No.
There's not that much embarrassing. Like, they're weird, but they're not embarrassing.
All of them, every single scene is kind of embarrassing.
It's weird, but not embarrassing. You don't even know if you didn't finish it.
Oh, didn't
I usually don't eat it.
So
All
weird.
right.
I couldn't.
No, but I do want to research.
Okay, the next part, we finished the nucleus and the ribosomes. The next one is the endomembrane system. Endo just means inside. So we did the cell membrane outside. Now it's the stuff inside the cell that has a membrane. Most of it is exactly like the cell membrane. So the phospholipid bilayer, it is the same thing on the outside. And you'll see that that's important that they're all the same. The endomembrane system has 4 components. The first one, if I remember what's going to appear, is one that acts differently, like we said, like you saw in the animation. You know, the nuclear membrane. Things don't come in and out the same way. They have their own gateways, those nuclear pores, and a gatekeeper there to make sure exactly what's coming in and coming out. The endoplasmic reticulum, which they're telling you that from now on they're just going to call the ER. The Golgi apparatus, sometimes it's called the Golgi bodies. It doesn't either, it's the same thing.
Your fish isn't doing too good.
Yeah, I don't know. They look weird right now.
The bass just running into the,
Why is it a dead fish in the weeds right there? He just flopped over.
like one of the little minnows.
Yeah.
Yeah. A lot of times because the bluegills take a bite out of it and then don't eat the rest of it, and it dies with the bite out of it.
I remember the one fish where his eye was gone. He was still swimming around. Just living.
Don't bring that up because that was somebody's lab fish. It's hard on them. And membranous sacs called vesicles, and most of us have some word that, even though we know how to spell it, we always want to spell it the wrong way. Vesicles is one of those. For me, I want to put V-E-S-S, but there's only one. But that is, so if you ever see I've written it or something and I put 2 S's, I know there's only supposed to be one. It just looks right with 2 S's to me.
Right.
Sounds like Coach Miller. All right, this system compartmentalizes the cell, and you're going to see as it changes, it's very fluid when you're going to see one of the animations. And one of them, I think it's nice because it's not an animation as far as somebody drew it. It's just time-lapse photography so that it goes a little bit faster, but it's actually part of this system. As your little workers, your employees are building the endoplasmic reticulum. Compartmentalizes the cell and carries molecules between components of the system. So it does some work inside in changing, for example, the proteins or lipids. Lipids are just, they're fat components, but they're not, most lipids in your body are good, they're necessary. So if we use fat, we just always think of fat as a bad thing. Lipids, not all fats are bad. We actually need to eat a certain amount of fats because we need them. The problem is we really like fatty stuff, and so we eat way more than what we need, the minimum requirement, and that's when it becomes bad. But so we never have, nobody ever wants you to cut out all fats out of your diet. It's just that we are so quick to eat fats that we don't have to really worry about not getting enough fat. We're right away to the other side. One strip of bacon probably does this for several days of the fats that we need. But who doesn't like bacon, you guys?
I don't like bacon.
Yeah, I would say some of you don't even go to church, so you probably don't like bacon either. But all right. The ER is a complicated system of membranous channels. And those channels, whenever it says membranous, it means that just think of those phospholipid bilayers, the polar, and they usually make them red, or in this case, a lot of the times they make them blue and green, kind of a turquoise color. And then the two little tail things look kind of like seaweed on the inside. Anytime it says membranous, you think of that phospholipid bilayer. Channels and flattened vesicles called saccules. It's kind of like taking a balloon and just squishing it flat, so you have this flat channel that's in between. Two types of ER. There is the rough ER, and it's studded with ribosomes. You're actually going to see a real scanning electron micrograph of it rather than just a drawing of it. You'll see that it actually looks like it has that. You won't see that. You won't see the endoplasmic reticulum with our microscopes at 400 times, which is our max. You're not going to be able to see that much detail. And the rough ER synthesizes proteins because it has ribosomes. That's what ribosomes do. Anytime you see ribosomes, it's going to be dealing with making proteins because that's what they do. Hopefully you get that down so when the organelle quiz comes up, and as soon as you see ribosomes, synthesize proteins, that those two always go together. And these proteins are packaged in transport vesicles, which that means we get to watch a animation. But we'll finish this first. We just have to talk about the smooth ER first. Smooth ER synthesizes lipids. They're also packaged in those same transport vesicles. Now the transport vesicles have the same double layer of phospholipid on it. It's exactly like the cell membrane, just like the endoplasmic reticulum, just like what surrounds the Golgi apparatus or Golgi body, which, ladies, how much would you have to love some young man before you would change your last name to Golgi? Because that's actually named after the Italian scientist who discovered them. Madison Golgi.
No,
that
you're
is
taking
not
my
it.
last name. That's not happening. That's such
Whoa.
an ugly last name. Golgi? No. Pinkerton's fine enough.
fine enough.
I like that last name.
Stop it.
Yours is basic. Shut your mouth.
Whoa.
Hey,
Hey.
that's his
Exactly.
Exactly. How many other people in this school do you think have the last name Schmidt?
Well, you know, it's,
8, 10 more.
well, there, I guess there always were quite a few, but they were all my kids. Now suddenly there's a whole bunch of them, and I'm not related to any of them.
They're on my
Well,
siblings.
more than I'm related to most of you. Although with a name like Schmidt, I'm sure anything like that, it wouldn't take too
Yeah.
long if you got somebody who was really good at the whole family tree thing. And there would be somehow your dad and my dad somehow would be related somewhere. But all right, now we're gonna watch this. And I don't remember which one this one is. Trafficking. That didn't sound good.
They spell
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. A principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, they are modified and packaged into vesicles. Because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components. The cis cisterna nearest the endoplasmic reticulum, or ER, the medial and trans cisternae, and the trans-Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them.
Okay, these are transport vesicles, and you're going to see that when they come over here, they just kind of fuse. It's kind of like when you have soap bubbles, and you blow a soap bubble, and you catch one on the end of the thing that you blew. It goes up, and you catch it, and you can catch another, and it looks like a snowman. It's got one on top of the other, and then all of a sudden, they just meld together, and they just have one big bubble. Then the outside of one and the other, they just join together and you just have one big one. That's the same thing as this, and that's why it's important that this has that same phospholipid bilayer as this does, as what the Golgi or the ER does, and you can see that it's studded with all these ribosomes. So this is the rough ER as they go across, and it's the same thing cell membrane has when it gets there.
Translated proteins are encapsulated in vesicles in the ER. A group of these vesicles fuse, and these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cis cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans-Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell.
That's coming.
The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
this is made by North Dakota State University. I was born in North Dakota, that's where my mom was from, like North Dakota, but it's a state university. But that looked an awful lot like design, where it went through and each one of them was changed in a way that said where it was going. And then at the last, the trans cistern, it actually was sorted just like in a post office. That whole sorting, I don't know that nature can just do that. Hey, let's get all these that are, they're not colored like that. All the yellow ones together and all the red ones together and all the purple ones together. They aren't colored, but just the whole organization of putting them all in one spot and then sending them out with the label on it that will send it to exactly the right spot. Sounds an awful lot like there was some programming that was done and design. I don't know how you can say that nature just by accident started sorting them. Nature doesn't sort things. Nature makes random piles of things. If you go through Fontenelle Forest and as the leaves come down, they're just randomly everywhere. They're not in piles. The sticks come down and they aren't put in little stacks according to size or anything. Trees just fall and they kind of decay. There is no order in what happens just by natural laws. There has to be something else that makes the order. So I think you can see order through all of this. Endoplasmic, now this is obviously the drawing. You can see and I can't raise this part up from this one, but these little lines here are the endoplasmic reticulum, and you can see all the little dots on the outside of them. Those are all the ribosomes. Over here there aren't the dots, so this is the smooth ER where the lipids are made. This is where the proteins are made up here. I think this is where you actually see them being built and taken apart. Yep.
The endoplasmic reticulum is a highly dynamic network of interconnected tubules that spans the cytosol of a eukaryotic cell, like a spider's web. The network is continually reorganizing, with some connections being broken while new ones are being formed. Motor proteins moving along microtubules can pull out sections of endoplasmic reticulum membranes to form extended tubules that then fuse to form a network.
These are your little workers that you have. We're going to meet them here in a little while, probably tomorrow. Okay, Golgi apparatus. And you've already seen part of what it does. That was those things, the different layers that it went through. Numerous flattened saccules, those were the cisternae that they went through. And usually when it's drawn, it looks like a stack of pancakes. Receives the vesicles from the ER, just like you saw in the animation. And it packages them in new vesicles. So they come in vesicles, they get changed, and then they get put back into new boxes and get shipped out.
And then it doesn't arrive till Tuesday.
Well, actually, what was scheduled to arrive by Thursday, now I just got an update here before school is coming tomorrow. So, yeah, sometimes they get ahead of schedule.
Very good. Where's it supposed to come from? Then, find out. Now they put you back.
But they do. Mine didn't seem to be affected, but each one of them comes with, it used to be with the COVID-19, because of that, the schedule might be off. Now it's blaming every hurricane that comes through might delay your thing. They always have some reason that we might not get it there on time.
It's been 5 days.
What is it that's coming?
Just wires.
Yeah, for his speakers.
Oh, okay. Monster wires? No.
Yeah, for his 30 inch.
No, that's the actual
No,
brand.
it's only 8 days.
Okay. The Golgi apparatus directs the new vesicles to the location intended for the protein. I didn't type these. This actually came with the secular evolutionary textbook. But when you start to give instructions, directions, that is always information, and information has to come from intelligence. Nature doesn't come up with that where all of a sudden it's writing on there, hey, take this to another place, and you're going to see that it's not just, well, it sends out and they have so far, it just kind of floats to where it's supposed to be. You actually have workers that read the label and then they carry it for you to where it's going to go. So they have to be able to be given that information and to understand it and to follow through.
Hey.
I looked around. Usually it's Madison.
3 rows ahead, so yeah.
Oh, it takes a while for the light to get there? At 186,000 miles per second.
How do you just know that?
He's a science teacher.
Yeah, but
How
like,
do you not?
Yeah.
300,000.
it's just such a
Maddie, you're
science.
weird for not knowing that. Like,
It's just
So
so
weird.
precise, and I would just like forget. I don't know. I would probably get like
the speed of light.
I would probably get the 3 right, and then I'd probably add a few more digits.
It doesn't matter. Yeah. People would believe you. That's
That's
all.
true,
If
you
you're
have to
gonna
say
be,
with confidence.
if you're gonna, yes, if you're gonna be a science teacher, you just have to say whatever you're gonna say with confidence, and people believe you.
That's expensive.
watch everything you're teaching us is a
Why do
lie.
you say that all the
Yeah,
time?
but by the time we find out,
Yeah, I know, because I got it from
are we
Mr.
good?
Schmidt.
All right.
Mr. Schmidt told me that last year.
All right, so this is a very quick just drawing of what you just saw. The rough ER is going to bud off these vesicles and has proteins in here that are made from all these little red dots on here. Those are the ribosomes. Make the proteins, and they come and they attach to the Golgi apparatus, and as it goes through, they get changed, and then they bud off here, and if they go to the cell membrane here, this red part is the cell membrane on the outside, they actually just, like the soap bubbles again, they just come up against it and they fuse with it, and then the vesicle becomes part of the cell membrane because it's made of exactly the same things, and suddenly whatever was inside just gets pushed out of the cell. Now that would make the cell get bigger and bigger, but the whole time there are vesicles that are coming in and budding off the cell membrane as well, so it stays the same. It goes through, it's in equilibrium. Some are joining it, some are coming and budding off of it. So the cell stays about the same size, but that whole budding process is how we transport stuff in and out. Now, some of them make lysosomes, which will be another organelle we come up with here, and that's kind of has a lot of different jobs, kind of the police, the recycler in there. And if the cell decides that these are bad things, then the police find it, and there isn't much of a trial or anything. The bad things are executed right away, which it's not quite as violent as what the animation you're going to see, but if you were making it, you would do the same thing just because you could. Usually there is on these. If I could find an animation, I put it in there. There's the name Camilo Golgi.
Camilo's compensating that.
Yeah. All right, and maybe, you know, now I don't know if
But he's
in Italian, I don't know if the LL is still an L, or if it's like Spanish and it, yeah, I want to say the L. Camilo, I want to say the L. Golgi. Also called the Golgi body, Golgi complex, or dictosome. Let's not go that far. Now again, this is not by a design organization, and yet you're going to see design in this. There's really no way to look at this and think, oh, that just happened by chance. There's the nucleus, endoplasmic reticulum. This is the Golgi apparatus, kind of a stack of pancakes, Cue the exciting music. I don't think your cells actually come with the soundtrack. So.
That'd be really funny. Oh my word.
Hey, extra for microscopes with little speakers.
Yeah.
The yellow thing is a protein. It's inside. They're just showing you what's inside those vesicles. As they come from the rough ER, and then they come out packaged. This looks a little bit like design here, where it gets packaged and then a label gets put on it and then it gets sent out. Yes, they keep doing the same thing over and over. But if you came up with that animation, you would replay it several times too. But it does. Oh, yeah. Nature does not do this by chance, by random, and putting the labels on and stuff. That is information that can come only from intelligence. And now they're actually showing us, like UPS or FedEx, where it decides even which direction it's going to send it out, which set of rollers there it's going to go down. And then when it goes, here it comes to the cell membrane, it fuses and it just sends this stuff out. That one was waiting for just for traffic to clear. All right, here's one, a lysosome that we did, and you saw something come in and it's going to execute that, and yet nothing really explodes inside your cells, but it's much more exciting if you're making an animation. Let's have it actually explode.
I like the theme music.
I like Kronk, it has its own thing. All right, this is the lysosomes. We'll just get through this one. Lysosomes are Golgi. That was the last one that you saw, the one that went out and it didn't go anywhere. It kind of just hung out there. That was the guard, the cell guard, security, waiting for the signal that, hey, we're bringing in some prisoners, and it's going to go over and digest whatever is in there, not blow it up, but digest it. And it will actually take a lot of the things that are meant to harm you, it just takes apart, and then the body reuses it, because it's probably made out of the same kind of proteins, and it just recycles it and uses it for whatever the cell needs to build. Sometimes it doesn't digest it. Could also go through apoptosis, which you have 100 million. Well, I wish I'd say that, because we would probably, if we gave Simon $100 million, he'd probably build us a football field somewhere.
Yeah.
But cells, so when one wears out, or parts of a cell wear out, they just recycle it through this. And now if your body decides to make a bunch of lysosomes and it completely destroys a cell, it's not like you're going to start to shrivel or you're going to even feel it or notice. You've got plenty of them. It's not going to bother you at all. If it becomes inefficient, then we're just done with it. All right, we will pick up from that mañana.
Are you warm?
You fit it. You don't fit it. But you fit it all. But like you fit it. Maybe go city girls over again.
I should do it. Like all the What? What?
So you guys practicing for a little bit and then eating a team meal? It wouldn't do to eat and then go do.
Yeah, it's only service.
Why?
Oh, no, you're not. You're perfectly sorry. I really did.
Because if I pass, I'm gonna drop you.
That's a funny.
I'm hungry.
I don't want to.
Recess.
Nice job. Yeah.
Yay.
Except it's cold
We had
out.
fun
We talked
recess
about how you
recess.
complain about
Cody,
having to
but
go.
we have-
In Mr. Game Master Theory, I stuck my tongue out at
That's
Kate.
I know
you would.
And then I
All right.
lied.
I know you did.
He wouldn't lie about that.
I told Coach after that. I was
Guys.
like, I'm
You're on film.
It's okay, they can hear that.
Emily will know. That's the only person. Oh no, Axel too. Okay, this is where we were yesterday. It is one of the parts that come off of the Golgi apparatus, the vesicles that come off. So they are Golgi vesicles, and they bud off with special enzymes in it that act as two jobs. They can be the security, and they can destroy foreign material, like the animation had. It doesn't actually blow up in there. That would be hard on the cell, but it will dissolve them, and most of the time it can actually take them apart to the stuff that it's made of, and then use that in the cell, recycle it, or they are just straight recyclers. Now here they talk about that. I think the animation is going to talk more about the recycling. If there's a part that's worn out, I think in the, what is it, in the animation, I think it's the mitochondrion, that stops working. It's just not efficient anymore. So the cell is going to need to build another one, but it can reuse the parts from that. So it will just take apart the old mitochondrion and into its basic pieces, put it back together as a new one, and that, but recycle, so we don't have to come up with new materials all the time. So it does security guard and recycling are its two jobs. Now the apoptosis is where it just destroys a whole cell because the cell isn't working anymore, and we'll talk about that later. A lot of times it's when the DNA gets messed up, because that's a danger to us if you've got a cell with bad DNA. That's how a lot of cancer starts. Cancer, we're going to find out several chapters down the road, is just it's a disease of mitosis, of cell reproduction, where it just keeps, the cells don't do anything, but they just keep reproducing, and so you get a bunch of massive cells, which we call a tumor, and it starts to take over that area and it's hard on that thing, and it stops whatever organ from working like it's supposed to. It can be very dangerous. So the cell, will actually, if the DNA is bad, just kill that cell. Again, you've got a lot of them. 1 cell gone, 2 cells gone, 1,000 cells gone. That you're not going to notice it. It's not going to be like all of a sudden you shrink a little bit because cells are disappearing. We'll make more. All right, and that is green. And I think this is the. I would like to.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
That's it. That's it. That's what it does. You now know everything you need to know about lysosomes. All right, vacuoles. Vacuoles are a simple part of this, but they're very important. They're just sacs in different types. Animal cells have much smaller vacuoles than what plants do. Again, we talked about it, and you'll see, because the plants have to store water because they have to wait for either you to water them or it to rain, whereas most animals have a way of going and procuring new water supplies. Now, I don't know why they put it this way. Vacuoles have specialized function in all cells, but plants are different because there's one central vacuole that is the kind of the water tower for the cell, and it also is part of the structure. There's quite a few things. We'll talk about it when we get to the dissections, and hopefully you are still here. We missed it last year, that part, but earthworms have a hydrostatic skeleton, which means that the water is actually what gives them. You need some money. There's some money right there. And that's why if an earthworm thinks that it's really a good idea to start to crawl across the sidewalk, or even worse, your driveway, you can go ahead and find them. We'll all sleep better knowing that they have been recovered off the floor. But as they go across, and if they can't get to the other side and the sun gets out and the robin doesn't get them, then you see them start to shrivel up. And the reason they shrivel up is because as the water evaporates, they lose the pressure that's in there. The water pressure is what makes them look like and work like a worm. Once they lose that, they can't move anymore. I don't know what we do when you're not here tomorrow. She is entertaining. But they can't move. Once they lose that water, there's no pressure. They can't stretch out and put those little hairs that they have. They actually have like little claws that they can put in. They're just hairs, but you can actually feel them part of the things with, before we cut the worms up, I will get you an actual pet earthworm that you can actually feel, and you can feel the rough part. Oh, you can feel the hairs that are there, and it reaches out and it grabs whatever, and then it pulls the backside with it. Come on in, Mr. Parson. Olivia, go tell him to come in. He thought we were doing something for him. Go chase him.
Mr. Parson. Mr.
Got a whistle. This too is on tape.
That's kind of cringy, not gonna lie.
So bad that fish.
Hey, Ms. Schmidt.
You are always welcome.
I was looking for JC,
See, she's right behind you.
Yep, I found her.
There she is.
Didn't want to interrupt class, but looks like I did.
It's all right. It's on film, so it'll be there for posterity.
And for what it's worth, your brother and sister-in-law are here.
All right.
Just so you know, be updated.
So if they don't have the water in there, they start to dry out. They can't reach out, grab it, and pull anymore. They're stuck there. It's not that they just get tired or in the middle of it. Once they lose moisture, they have no way of moving anymore. So they're just there waiting for a robin to come get them or to just dry up. And after they're dried up, then only dogs will come and eat them. And they do.
That's gross.
It might be, yes, it is, but I think it's gluten-free. In most organisms, vacuoles can store nutrients, ions, and other molecules. But we don't store water as much. Our water is stored in the plasma of our blood if we need it. That's the way we control how much water is in there. If you've been drinking and hydrating yourself, you've got plenty of water that it can be just distributed wherever it needs. If you haven't been drinking enough, then you start to get dehydrated, you get headaches and stuff, and a lot of that is just because there's not enough moisture for all of the cells there. But nutrients, we store nutrients. They don't dump all of the, let's say that Madison goes out for ice cream and she eats, you can eat ice cream, Keely can't, but you can. She eats ice cream and all those nutrients, we'll just call them nutrients, are in there. The body doesn't dump all of those into the cell and just burn them all up right at the time. One, we would overheat because burning up the stuff gives off energy, gives off heat. So he is here. So we do, it will take them in, thank you, and then just put them in the pantry. And the vacuoles act as that pantry. And then in case we actually go for a while without eating anything, then they can go to the pantry and pull those nutrients, the ions, whatever we need out of the pantry and use them. If we do eat again, if we don't actually give them a chance to do that, then they eventually get pulled out of the pantry and they get taken to the liver to go into long-term storage, which fills up quicker than what we'd like to. I think now a picture. Here is an animal. Well, when I was your age, it was an animal cell. It's now a protist. That looks like a paramecium. And these big, they look like suns, are the contractile vacuoles, and they store stuff. And the arms, I think, are just ways of dispersing whatever's in there out. And in the plant, this is a plant cell. You can tell here's the chloroplasts. We're coming to those. They have this large, looks like a swimming pool here in the middle, more like a reservoir. If you've been down by Dodge Park, if you've played soccer or softball or something, Dodge Park down by Florence, and if you drove from there to the airport, you go past Omaha's water treatment plant, the one that's on the Missouri River. There's another one that's on the Platte River that's down by between Omaha and Plattsmouth. But the one on the way to the airport, between Florence and the airport, they have a bunch of just water, looks like huge swimming pools, that where our water, our fresh water is cleaned and it's settles there for a while, and all the stuff falls out of it, and then it goes through and is chemically cleaned, so that we have it. And we have great water. Nationally, when they rank water, Omaha always ranks very high. We don't have a lot of, I mean, the Missouri, I wouldn't go dip a cup in the Missouri and drink it. It's very dirty, but most of it is just dirt and mud and stuff like that, which is really easy to clean out. We are getting more and more, just as the farmers use different things, more and more of the chemicals. That's the part that's harder to get out, is the farm chemicals that wash down off the fields and they eventually get into the river. That takes more work to get them out. Just the dirt itself settles out very quickly. But this looks just like that. And again, when we look at them, you aren't going to see this. We go to 400. This is at almost 8,000 magnification. We aren't going to be able to see that. You'll actually be able to infer where it is, because there won't be any chloroplasts around. There will be the empty spot where there aren't any chloroplasts. Neat. At least neat. All right, that's the end of that section. We just have two more of the energy related. We will do that tomorrow because my brother's here.
Yes.